In the vast universe of drug discovery, a revolutionary technology has emerged, promising to reshape the landscape of medicine. This groundbreaking approach is called PROTAC, or proteolysis targeting chimeras. PROTAC technology was born at the beginning of the 21st century as an innovative drug discovery strategy aimed at treating diseases through the selective degradation of specific proteins. Conventional small molecule inhibitors work by blocking enzyme or protein activity. But this approach has limitations when dealing with certain protein targets, such as non-druggable targets. PROTAC technology was first proposed by scientists such as Cruz and Deshays in 2001 and has rapidly evolved with the progress of research. Evolved rapidly as research progressed. It is groundbreaking in that it enables protein degradation rather than simple inhibition by directing the target protein to interact with the intracellular ubiquitin proteasome system. With continuous improvements in technology and chemical synthesis methods, PROTAC is now an important tool in the development of drugs for a wide range of diseases, including anti-cancer and neurodegenerative diseases. As researchers delve deeper into the intricacies of PROTAC design and optimization, the therapeutic potential of this revolutionary technology continues to expand, offering new hope for patients worldwide. The ongoing research and development in this field promise to bring forth a new generation of treatments that could change the face of medicine as we know it, providing solutions to some of the most challenging health issues of our time. PROTAC molecules, despite their complex mechanism of action, are deceptively simple in their overall design. The efficient design of PROTAC molecules relies on its three core components: target protein binding structure, E3 legacy ligand, and connecting chain. Each part plays a key role in ensuring that PROTAC achieves efficient and specific targeted degradation. The target protein binding structure is the part of the PROTAC molecule that recognizes and binds to the target protein. It is usually composed of small molecule compounds, peptides or nucleic acid fragments, and can selectively recognize and bind to the target protein. For different target proteins, the selection of ligands must ensure high affinity and specificity to ensure the formation of a stable ternary complex. The main principles for designing target protein binding structures include ensuring their high affinity, specificity, and stability in the cellular environment. The challenges faced when selecting target binding modules include how to overcome the flat areas on the protein surface that are difficult to bind, and ensuring that the selected ligand does not interfere with the ligand part that binds to the E3 ligase, thereby affecting the stability and function of the complex. E3 ligase ligands anchor PROTAC to ubiquitin ligases and promote protein ubiquitination. Common E3 ligase ligands currently include von Hippel-Lindau, Cereblon, etc. These ligands have shown good degradation activity in many PROTAC designs. The selection criteria are mainly based on the widespread expression of E3 ligases, the binding ability of target proteins, and the availability in cells. In addition to the common VHL and CRBN, researchers are exploring other E3 ligase ligands, such as MDM2, IAP, etc., to broaden the scope of application and degradation ability of PROTAC. Different E3 ligases have different substrate specificity and intracellular expression patterns, so choosing the right E3 ligase ligand in the design can significantly affect the function and applicability of PROTAC. The linker chain is used to connect the target protein binding structure with the E3 ligase ligand. Common chemical structures include polyethylene glycol groups, alkyl chains, etc. PEG-based linkers generally provide higher water solubility and flexibility, while alkyl chains provide higher hydrophobicity and rigidity. The length and flexibility of the linker chain directly affect the formation, efficiency, and stability of the ternary complex. Shorter linkers may limit the binding of the target protein and the E3 ligase, while overly long or overly flexible linkers may reduce the stability of the complex. Therefore, optimizing the length and chemical properties of the linker chain is an important step to improve the activity of PROTAC. The spatial layout of the linker chain is crucial to ensure effective contact between the target protein and the E3 ligase. Studies have shown that appropriate spatial positioning and linkage angles help form stable complexes and promote the ubiquitination process.
PROTAC is a class of bifunctional small molecules that achieve selective protein degradation by connecting the target protein to the E3 ubiquitin ligase. Through specific structural design, PROTAC forms a ternary complex between the target protein and the E3 ligase, triggering the ubiquitination of the target protein and marking it as a target for degradation, which is then recognized and degraded by the proteasome system in the cell. The design concept of PROTAC is based on its bifunctional structure. Each molecule includes the following two core binding structures. The target protein binding structure is the part that recognizes and specifically binds to the target protein, ensuring that PROTAC can bind tightly to the desired target protein. The E3 ubiquitin ligase binding structure is responsible for binding to the E3 ligase. Common ligands include molecules that bind to E3 ligases such as cereblon and von Hippel-Lindau. The two structures are connected by a chemical linker, allowing PROTAC to bridge the target protein with the E3 ligase to form a stable ternary complex. When PROTAC binds to the target protein and the E3 ligase, the ternary complex formed enables the E3 ligase to attach the ubiquitin tag to the target protein. The ubiquitin-tagged protein is then recognized as a degradation signal and transported to the 26S proteasome for degradation. This process is efficient and repeatable and can achieve continuous removal of the target protein. Compared with traditional small molecule inhibitors, PROTACs have the following significant differences and advantages. Traditional small molecule inhibitors prevent their function by binding to and inhibiting the activity of proteins, while PROTACs eliminate their biological functions by degrading proteins. PROTAC can more effectively deal with drug resistance problems caused by protein mutations or overexpression through protein degradation rather than simple inhibition of activity. Because PROTAC degrades the target protein, the effect is often longer lasting than simple inhibition and does not require continuous high concentrations of drugs to maintain the inhibitory effect. PROTAC can target undruggable proteins that traditional inhibitors cannot effectively act on, expanding the potential scope of drug development. These properties of PROTAC make it a promising new tool in modern drug development, especially in the study of cancer, immune diseases, and other complex pathologies.
In PROTAC molecular design, key strategies include chemical modification, linker chain optimization, molecular dynamics simulation, etc. These methods help improve PROTAC's targeted degradation efficiency, affinity, pharmacokinetics, and other properties. Below, we will introduce the detailed optimization strategy. Optimizing the target protein binding structure of PROTAC molecules and E3 ligase ligands through chemical modification can significantly improve their affinity and selectivity. This process usually involves identifying and modifying the key interaction region between the target protein and the E3 ligase, allowing PROTAC to efficiently connect the target protein to the E3 ligase and trigger the ubiquitination and degradation processes of the target protein. Optimizing the target protein binding structure can also improve the selectivity of PROTAC for specific targets, thereby reducing the impact on non-target proteins and improving the overall therapeutic effect. The length, polarity, and rigidity of the linker between the target protein binding part and the E3 ligand in the PROTAC molecule will all affect the conformational stability and pharmacokinetic properties of the molecule. Appropriate chain length helps optimize the spatial arrangement of the molecule between the target protein and E3 ligase and enhances its binding efficiency. Polarity and rigidity will affect the membrane penetration, stability, and distribution of the molecule in the body. By adjusting these parameters, the bioavailability and half-life in vivo of PROTAC can be effectively improved, and its drug performance can be improved. Using molecular dynamics simulation to study the conformational changes and target binding efficiency of PROTAC molecules in vivo is an important means to optimize design. Molecular dynamics simulations can reveal the structural dynamics of molecules in different environments and help predict their binding efficiency to target proteins and E3 ligases. This method can further guide the structural optimization of molecules, allowing PROTAC to more efficiently degrade target proteins in the body and improve drug efficacy. Optimizing the linkers in PROTAC molecules to improve their stability and efficiency can be carried out from the following aspects: introducing polar curing groups, changing the length of the linkers, introducing rigid structures, introducing oxygen atoms and other functional groups, using biological physical techniques, monitor TC stability, enhance TC synergy, computational modeling prediction and design, and optimize oral absorption and overall bioavailability. By combining the above design and optimization strategies, the activity and selectivity of PROTAC molecules can be significantly improved, making them a potentially effective tool for the treatment of refractory diseases.
The synthesis of PROTAC molecules requires the covalent connection of the target protein binding module and the E3 ligase ligand through a linker to ensure that the molecule has an effective target protein degradation function. Below, we will introduce the commonly used techniques, key reaction steps, and industrial challenges in PROTAC synthesis. The synthesis of PROTAC molecules relies on the connection of the target protein binding module and the E3 ligase ligand through a stable covalent bond. Generally, a connection method based on amide bonds, ester bonds, or click chemistry is selected to ensure the stability and controllability of the connection chain. The synthesis steps of amide bonds and ester bonds are relatively mature, while click chemistry, such as the cycloaddition reaction of azide and alkyne groups, can efficiently and selectively generate covalent bonds under mild conditions. In addition, the selection and design of the linker chain will affect the activity and specificity of PROTAC, so special attention should be paid to the optimization of its structure and length during the covalent connection process. The synthesis of PROTAC generally includes the following steps: precursor molecule synthesis, linker chain modification, molecular splicing reaction, purification and identification, etc. First, the precursor molecules of the target protein binding module and the E3 ligase ligand are synthesized separately. These precursor molecules are usually prepared through multiple step reactions and need to be purified to ensure high purity. The appropriate linker is selected according to the characteristics of the target protein and E3 ligand and is functionalized, usually including modification of carboxyl, amino, or azide groups. The target protein binding module, E3 ligand, and linker are covalently linked under appropriate reaction conditions. The key reaction conditions for this step include temperature, pH, catalyst, and solvent selection. Ensure mild reaction conditions to maintain the activity and structural integrity of each module. Purification by column chromatography, HPLC, or mass spectrometry techniques ultimately yields a high purity PROTAC finished product. Its structure and purity are then identified by nuclear magnetic resonance, mass spectrometry, and infrared spectroscopy. The industrial production of PROTAC faces some unique challenges. Due to the large size and complex structure of PROTAC molecules, reaction conditions are prone to change during scale-up, which may lead to reduced yield or increased byproducts. PROTAC molecules usually have multiple chiral centers and long linkers, which makes it difficult to ensure the stability and consistency of each batch of products. The synthesis process of small molecule compounds may not be applicable in large-scale production, so it is necessary to develop stable and low-cost production routes. In terms of quality control, the purity and stability of PROTAC need to be strictly tested by HPLC, LCMS, and other methods to ensure that each batch meets the standards in terms of targeted activity, purity, and impurity content. In addition, the integrity of the connecting chain and each module is verified to ensure the activity and selectivity of PROTAC. By optimizing the synthesis method, improving the reaction conditions, and establishing a strict quality control system, the industrial production of PROTAC can be gradually stabilized, laying the foundation for its application in the fields of medicine and biotechnology. The diversified application potential of PROTAC technology is being widely explored, and it has shown significant advantages in anti-cancer, neurodegenerative diseases, antiviral treatment, and other fields. We will explore the main development directions of PROTAC in various major application fields. In cancer treatment, PROTAC technology inhibits the proliferation of cancer cells by targeting and degrading oncogenic proteins. Traditional targeted therapy drugs usually work by inhibiting protein activity, while PROTAC can directly mark the target protein as a target for degradation, thereby completely eliminating its expression within the cell. This approach can overcome the drug resistance of target proteins and is particularly suitable for oncogenic proteins that lead to cancer development due to overexpression or mutation. For example, PROTAC has been developed to degrade and inhibit proteins related to cancer cell proliferation, becoming a potential weapon for precision tumor treatment. In the field of neurodegenerative diseases, PROTAC technology provides a new solution for clearing abnormally accumulated proteins in diseases such as Alzheimer's disease and Parkinson's disease. Such diseases are often triggered by protein misfolding and accumulation, such as amyloid beta in Alzheimer's disease and alpha-synuclein in Parkinson's disease. PROTAC can target and degrade these abnormal proteins, help restore normal functions of the nervous system, and slow down disease progression. This technology not only has selectivity for specific pathological proteins, but also reduces the side effects of traditional drug treatments and has broad clinical application prospects. PROTAC shows important application potential in antiviral therapy. By targeting and degrading key proteins in the viral replication process, PROTAC blocks viral reproduction. For example, certain PROTAC molecules can specifically degrade proteins related to viral invasion and replication, thereby reducing the survival and spread of viruses within host cells. This technology has potential in the treatment of HIV, HPV, and other viruses, and provides a new strategy for the development of antiviral drugs. In addition to the above three major fields, PROTAC technology can also be applied in the following potential directions. By regulating the degradation of key proteins in the immune system, it can effectively enhance or suppress immune responses and provide support for immunotherapy. PROTAC molecules can selectively degrade proteins related to bacterial survival or virulence, providing a new antibacterial pathway against drug-resistant strains. PROTAC can also achieve gene regulation by degrading gene expression regulatory proteins, bringing new tools for gene therapy and molecular biology research. Through in-depth research and exploration in different fields, PROTAC building blocks are gradually becoming a new means of treating multiple diseases and pushing drug development into a new stage of precise degradation treatment. PROTAC technology, as an emerging targeted degradation strategy, has broad application prospects in the field of drug development. Future development will focus on the following key directions to overcome the limitations of existing technologies and enhance the therapeutic potential of PROTAC. Most of the currently known PROTACs utilize a few E3 ligases, such as VHL and CRBN, but many disease-related target proteins cannot be effectively degraded by these E3 ligases. Future research will focus on discovering and developing more types of E3 ligases to expand the scope of PROTAC. By identifying the unique mechanism of action of different E3 ligases with specific proteins, researchers can design PROTAC molecules that are more suitable for specific targets, thereby improving the selectivity and efficiency of PROTAC. In order to improve the functionality and stability of PROTAC molecules, future research will focus on introducing more natural molecules and synthetic fragments as building blocks of PROTAC. Specific molecules in natural products may have excellent target protein binding ability or unique biological activity, while synthetic fragments can improve the stability and targeting of PROTAC molecules through chemical modification. Diverse building blocks can not only provide more flexible molecular design options, but also provide effective solutions for target proteins that are difficult to degrade. The clinical application of PROTAC faces challenges such as in vivo stability, bioavailability, and targeting specificity. PROTAC molecules are large and complex and are easily metabolized and degraded in the body, limiting their bioavailability. Future research will focus on improving the pharmacokinetic properties of PROTAC, such as optimizing the structure of the connecting chain to improve molecular stability and increasing the effective concentration of PROTAC in the body through new delivery systems, such as nanocarriers. In addition, increasing the targeting specificity of PROTAC will also be an important research direction to avoid potential side effects and improve therapeutic effects. Machine learning and artificial intelligence have broad application prospects in PROTAC design and optimization. By building an efficient algorithm model, researchers can quickly predict the bioactivity and stability of PROTAC molecules with different structures and accelerate the screening and optimization of candidate molecules. Intelligent design tools can use big data to analyze properties such as molecular conformation and target binding and provide more precise design guidance for PROTAC molecules, thereby shortening the R&D cycle and improving design efficiency. In summary, future PROTAC research will improve its application effect through the innovation of E3 ligases, diversification of building blocks, optimization of pharmacokinetic performance, and intelligent design. These cutting-edge developments will promote the application of PROTAC in the treatment of various diseases, gradually realize the transformation from laboratory to clinic, and provide new solutions for personalized and precision medicine.